Today we're pleased to welcome Lucia Jacobs of UC Berkeley, and she'll be speaking on chemosensory cognition and the evolution of olfaction.
Thank you. Well, it's a great pleasure. Thank you so much for inviting me. I've already had amazing conversations. I'd like to say, everyone, please just jump in with questions. I've started studying olfaction about a year ago, so I have lots to learn. And if you've got questions, they're probably really great questions I'd love to hear. So we'll just use the whole time mixture of talk and questions. So I always like to start with why, how to study behavior with my God, who is, sorry, I moved around, with an outline. This is what the talk's going to be. What I want to talk about is unpacking kind of a higher cognitive concept and suggesting today that it actually can be understood as actually quite a simple process using quite primitive processes. And that is the phenomenon of the cognitive map. So I'm going to start with a cognitive map, unpack it into its components, then turn to a paradox of the cognitive map, which is that despite the cognitive map being mostly characteristic of vertebrates, large-brained animals, it's now clear that the same mapping can be done by animals with very different, very simple brains. And I'm going to then address the paradox of the cognitive map with another paradox, which is the paradox of olfaction, the sense of smell. And I am going to think about the paradox of olfaction. And there are many interesting characteristics of olfaction. One is that it is similar in all animals, incredibly similar. And I mean all animals. And what I'm going to propose is that one paradox solves the other paradox. That the reason this thing that we think is so high cognitive, the cognitive map, can be done by animals with tiny brains is because both we and they have the same olfactory system. And in fact, the olfactory system is solving the cognitive map. And push it even further, the olfactory system is navigation. And we should be thinking about it as navigation. It's a universal mechanism for the cognitive map. And I'm going to discuss other people's data from mammals. And I'm going to discuss implications of this for brain evolution. And so, this is like I said, a new idea, and I'm exploring it. And today's talk is really just about the idea. It's with just a little smattering of someone else's data at the end. So, how to study anything? I always like to start with my hero Tinbergen. Is how to study behavior. We need to understand it at all different 4 levels of analysis. Actually, a lesson I learned both from Tinbergen and from Steve Glickman, which is its physiological mechanisms, its ontogeny, its ecological function, adaptive significance, and its phylogeny. And nowhere is that more important than for olfaction and the cognitive map. So what is the cognitive map? Well, it actually started as a very interesting kind of historical convergence in theory, where 2 men in the late 40s both conceived of this idea that a non-human could topographically map its space. And of course, an idea that was anathema to the behaviorist tradition of the time. And these 2 men were Edward Tolman at Berkeley studying the rat, arguing that rats represent the space of the maze as a topographic map, Across the Atlantic was Gustav Kramer, who was studying the homing pigeon, and exactly the same time in the late 40s and early 50s, proposed the map and compass hypothesis to explain how a homing pigeon could be released hundreds of miles from its familiar location and yet could beeline back to its loft. And in fact, what we're seeing now is that these two theories are now coming together. And we call it cognitive mapping in psychology, and we call it true navigation in biology. But essentially, we're talking about what looks like the same process, just under different different spatial scales. And the reason we can say that, one strong line of evidence is the neural basis of this, the cognitive map, is a structure which is the medial pallium, which is one of the four areas of the ancestral forebrain of the vertebrate. And the medial pallium is found in all vertebrates, and it's in all the vertebrates that have been studied, including fish, reptiles, birds, and mammals. It's required for making a spatial map. It's actually, this is the medial pallium of a skate, cartilaginous fish. Here's a bullfrog medial pallium. There's your medial pallium, and there's the rat medial pallium. And it's probably the one structure that has been studied, that has an interesting cognitive function that has been studied across so many diverse taxa. And it's also not only found in all vertebrates, but in reptiles, birds, mammals. The relative size of that structure can be predicted by the space use of the species, or the sex, or the age of the animal. Specifically, the flexibility in space use, shortcutting across a space. And this work, the neuroecology work, actually was built upon some remarkable work showing that individual neurons in the hippocampus, now the hippocampus is the mammalian homologue of the medial pallium. The hippocampus, it's the hippocampus in the bird, and it's the hippocampus in the mammal. It's called other things in other taxa. But individual neurons of the hippocampus encode specific points in space. And as a rat moves around a space, different neurons fire as it traverses different points in space. And so when this was then the hippocampal theory of the cognitive map, which was John O'Keefe and Lynn Nadell articulated this in the 1970s. And at that point, Tolman was finally vindicated that there was something to the cognitive map, and since then it's a minor industry in neuroscience, is how the hippocampus encodes space this way. Okay, so Francois Schenk and I addressed the question of how this cognitive map evolved with a theory we proposed in 2003, which is the parallel map theory of the hippocampus and the cognitive map. And it was the first hippocampal theory to ask, where did this structure come from? It's not where, and because it has, it's such a widespread and conservative function in vertebrates, what we proposed was you have to understand its function in terms of a naturalistic environment, such as this landscape, where you have a mixture of kinds of cues. You might have a cue such as that distant sun, that's moving across the sky, or a distant mountain range that is just being used as, ethologists say, a compass mark. It's just being used for direction, versus you might have landmarks that are local, that can be used to orient on a very fine scale to define locations. And so a typical environment, a natural environment where the hippocampus evolved, has this kind of wide range of different types of cues. And of course, in the what had happened is the hippocampus was only being studied in labs where they were small spaces with only landmarks, and also everything was very static. And so what we proposed, I won't go through the whole thing, but there was an enormous amount of disagreement in the field. And what we suggested is, if you take all these 2,000 studies at that point, 2003, and you break them up into whether or not the task is encoding a sense of direction or a sense of place, you could see that, in fact, different lesions of the hippocampus impaired the sense of direction, and others impaired the sense of place. And so we came up with this idea that there's actually two maps in the hippocampus, and that the cognitive map, from a cognitive point of view, unpacks into the bearing map, which is the kind of underlying graph paper of the cognitive map, which is encoded based on directional cues like that distant mountain. And in contrast, there are sketch maps, which are clusters of landmarks, local landmarks, which we call positional cues, which are being used for position. And it is then, these two maps are integrated, and when they're integrated, the positional cues, the small landmarks of the sketch map are recoded into the coordinate system of the bearing map. And then that is the reason you can shortcut between two positions, even if you have never, two landmarks where you have never been taught the route from one landmark to the other landmark, but because the two landmarks sit on a common coordinate system, you can simply make that calculation, that mental calculation, and know based on its XY coordinate position where you need to go to get to the other landmark. So if this is a theoretical suggestion that something like that would solve the mystery of animal navigation, how it is that something like a pigeon can be released hundreds of kilometers away and yet can beeline home, because there is a common coordinate system. There is some directional cue that is so widespread over the landscape that it can fly around as they do in a circle, and then pick up changes in this directional cue, a gradient, and know where they are in relation to home, and then home down the gradient to home. And this has been known, this has been recently shown in fruit bats navigating in the desert in Israel. Now we've got a mammal doing exactly what a pigeon is doing, released hundreds of kilometers from any familiar location, does this circling, picks up something, some gradient, and beelines home. You know, the not so secret punchline of my talk is that thing they're picking up is olfaction, is olfactory. Okay, so okay, this is that's the cognitive parallel map theory. This is the anatomical cognitive map theory, and this is everyone who studies hippocampus has their own cartoon image. This is Ramoni Cajal's image. This is what the hippocampus really looks like. It's beautiful and complicated. But what we realized is that the hippocampus can also unpack into two maps, into the more ancient bearing map, which is found in all reptiles, birds, mammals, specifically one subfield called the dentate gyrus. Sketch maps are found in the more derived part of the hippocampus. The hippocampus actually unpacks into two structures, the dentate gyrus and the Ammon's horn. They sit together like this. And so the dentate is the old one, and in fact, the dentate, lesions of the dentate impair the sense of direction. Lesions of CA1 impair the sense of place. And then there's a third subfield that sits between them, and that we've proposed is the integrator that integrates them into one cognitive map. And so that this is the, not important, but this is the bearing map circuit, which is these septal nuclei, and this is all the old circuitry, very found in all vertebrates. This is only found well developed in birds and mammals, and so that is and CA3 also is famous for its auto-associational loops. Anyway, the anatomy is consistent with this idea of two maps that are integrated like that. And this is what it looks like. Again, here's a bearing map where it's either you can have a gradient up to the top of a mountain or a polarized body of water. That anything that polarizes the landscape could be a gradient. These are visual gradients. And the sketch maps are the topology of small objects, positional cues. Even if they are identical throughout the landscape, it's their topology, it's work on CA1 function shows that the CA1 discriminates between, it's very sensitive to small objects and discriminates between these different the topologies of these different clusters, and then the integrated map is combines them. Okay, and recently there's been some very interesting work from Denise Mannheim-Vaughn's lab in Germany that she discovered and then discovered our theory afterwards, that if you have a rat exploring a rectangular arena and you that exploration activates different forms of synaptic plasticity, and specifically long-term potentiation, long-term depression, but it's basically just, it's experience-dependent plasticity, but the type of cue stimulates plasticity in different parts of the hippocampus, exactly as predicted by the theory. And so a directional cue, a tall object in the arena stimulates LTD only in dentate gyrus. A cluster of small cues stimulates LTD only in CA1, and both types of cues stimulate LTD in CA3, with the directional cues stimulating LTD in the connection from dentate to CA3, and the positional cues stimulating it in the connection from CA3 to CA1. And again, these cues are not how I would have designed it to test parallel map theory. This is all post hoc, but it just absolutely, it's very exciting. That's kind of independently this whole lab. Now they've got a series of papers on this showing this very specific, they're very excited because it's cue-specific plasticity in hippocampus, but it fits with the parallel map idea. Okay, so that's you have to know this because you have to understand where I'm coming from when I say parallel map. So now, as I said already, we have a problem. We have a problem, Houston. We have cognitive mapping without a hippocampus. And this has become a recent embarrassment to the field. There are two now studies in arthropods, insect, honeybee, now that we have harmonic radar, so you can track a bee across kilometers, which we never could do before. All the debate, do honeybees have a cognitive map, that's been going on since the 80s, has now been laid to rest, because when you have when you actually collect the data, they're doing beautiful shortcutting across areas they've never been before, just like just like a pigeon, over kilometers. So it's very clear that they are showing cognitive map-like behavior, and this has been the conclusion of the honeybee community. And recently, lobsters in a simulated magnetic sense have been showing a simulated magnetic field in the lab are able to point to and swim towards home, and these are spiny lobsters, which are Caribbean migratory lobsters, which move over very long distances. And they, in the lab, are showing what can only be defined. If this was a rat, you would have to call it a cognitive map or true navigation. So you've got two arthropods, a crustacean and an insect, but no hippocampus, obviously. So what do we do? Again, as I already said, what if we look at what is the neural common denominator between rats, pigeons, bats, bees, and lobsters, and there is only one, and that is the olfaction. And here is your nose, and it's inputting to the olfactory bulb, which is composed of glomeruli, which are tangled balls of neuropil. And that glomerulus, that glomerular structure, is found in all animals except for nematodes that have been examined. So here is the dotted line are your glomeruli. This is a mammalian olfactory bulb, and this is the functional equivalent in an insect, the antennal lobe of an insect. And this is just to point out that again, the wiring diagram of this first relay, the odorants are hitting the epithelium and then they're being sent to, this is the first relay, the glomerulus, and how that data is then collected and processed and sent on to the second relay is eerily similar. And it's not just to look at, there's beautiful computational models showing that the kind of information processing characteristics of these two systems are also almost identical. So, of course, the conclusion has been that's a good reason, because they're doing the same thing. They're discriminating odors, obviously. So, but I also want to show, this is Heather Easton's cladogram showing the olfactory glomerulus is present in all of these animals, everything but the C. elegans, crustaceans, insects, mollusks, bony fish, amphibians, rodents, humans. And so many people have been pointing this out for a long time, very notable neuroscientists and neurophysiologists and theoretical computational neuroscientists, because arguing that it's this, this incredible convergence. We can learn so much from it because it's so convergent. But again, the explanation is that we're all using the nose to discriminate odors. But there's this gadfly on the side who has been bothering everyone since 1970, who's Floriano Papi. And what he's an Italian ethologist who pointed out and suggested and hypothesized and tested the idea that homing pigeons actually use odors to navigate. And if any of you are from the field of animal behavior, you will know that this became such a bitter debate that many people just left the field of navigation because they couldn't deal with the mudslinging between the magnetic camp and the olfactory camp, because the magnetic camp was had the upper ground in 1970. I was trained in the magnetic. I actually studied biology under William Keaton at Cornell, which was the epicenter of the magnetic camp. So I of course dismissed this as an undergraduate. This is what you get for being dismissive as undergraduate. You're eating your words decades later. But in fact, now that the dust has settled, what it turns out is in Germany and England and upstate New York, birds are kept inside lofts, and they don't get the same amount of olfactory experience as they do in Italy, where it's beautiful and warm, and the lofts are on the tops of buildings, and they're open to the breezes, and it's much more natural for a rock-dwelling species like the homing pigeon derived from the rock dove, to be growing up with the breezes with all the odors. And in fact, the stronger the breeze, work has shown, the better they learn it too. So they're very sensitive to wind and odor. And when they cross-fostered Italian birds to indoor lofts and German birds to Italy beautiful rooftop lofts, you found that in fact the German birds would use olfaction to orient, and the Italian birds would use magnetic cues to orient. And so the story basically is you use the cue that best informs your cognitive map depending on what is most reliable where you grow up. And so if you want to use a cue that is spread over a landscape to solve that cognitive map problem, to choose the directional cues that you're going to use to build your bearing map, it'll be olfaction in Italy, and it'll be magnetic fields if you're raised inside in Germany. But the fact is, when the natural pigeon not only uses olfaction, but now there's been these beautiful double dissociation studies where they either cut the olfactory nerve, or they cut the trigeminal, a branch of the trigeminal nerve, which is necessary for encoding magnetic fields. And they just see what the birds do. And it turns out that a pigeon, so here's a typical pigeon orientation experiment where this is the loft and they're being released from these different locations, and that's a control bird, and you can see scatter, but they're basically all heading towards home. And here's a bird that's anosmic, so it's either had its nostrils plugged on the way out to the release site or at the release site, or it's got its olfactory cortex lesioned, or it's had its olfactory nerve cut. I mean, they've every possible, this was so, there's been so many studies to show that this is convergent studies to show that olfaction not only is important, but in these double dissociation studies, the birds with a magnetic lesion were basically unimpaired. Birds that had been raised in olfactory fields were unimpaired, losing magnetic fields, but birds who were raised in olfactory fields and lost the olfactory fields were highly impaired and could not just switch back to the magnetic. And it also turns out that the homing pigeon is for 2,000 years has been raised to competitively solve the cognitive map problem, and to do this, and it actually not only has a larger hippocampus than other pigeons of other pigeon strains, it also has a larger olfactory bulb than other pigeon strains, which is interesting, right? But you might think, yeah, well, that's just an olfactory specialist. But recently, Holland has replicated the same method, and they looked in New Jersey at the catbird, which is a little short distance migrant passerine. These are not navigation specialists, and they caught them right during the migration, and they did the same manipulation, either severing the olfactory nerve or severing the trigeminal nerve, and they found exactly the same thing, that the birds with the magnetic lesion could compensate, the birds with the olfactory lesion could not. And so that's a non-specialist showing the same thing. And this is also, you're seeing all the data practically on how vertebrates orient to odors. There's, because of the assumption that vertebrates use their nose to discriminate, it's really only within the bird navigation community that this hypothesis was proposed and has been tested extensively. But it's remarkable that it's so important to them, even given that obviously birds have less of an olfactory system than mammals. They're incredibly visual, et cetera, et cetera. This is kind of the last species you'd expect, the last group that would be using olfaction. Now, of course, olfaction in birds is becoming a very hot topic. They use it a lot more than we've known. So what I'd like to propose is, let's think about olfaction in a different way, a new paradigm. The presumed primary function has been discrimination, and the secondary function is you navigate using this in response to these odorants that you've discriminated. But what if we just reverse it? What if navigation evolved as a primary function, and in fact, you don't need that much discrimination, you just need enough odorants. You have to know the odorants that are distributed like olfactory fields, like magnetic fields, and you're orienting to that, and of course, then there's going to be selection for getting better and better discrimination. So this is the olfactory spatial hypothesis, and let's see how far we can take it. Before we go any further, a question, yeah?
Yeah, just a couple. I know for moths and a number of other animals, it's proposed that the olfactory sense, or the odor, is more or less a gating phenomenon. And so an organism will turn on basically some visual or a response to wind direction once it's received a given smell. And so I'm wondering if this could be the same, in that the chemical cue may be important in telling, let's say, a pigeon or something to look at the orientation of images across your retina to be able to distinguish this side from that side. So the chemical's important, but it's not actually navigating by the odor alone.
No, that's been proposed, and that's also being debated very much. That's been proposed by John Phillips, that it's a priming effect. But there's specific effects like pigeons who are raised in a loft with a novel odorant coming from a specific compass direction, and then they're given that odorant in an area where there are no visual landmarks they have in common. So they have no other method of orienting, and they're given that odorant that came from the north when they were at home, and they're given a very strong dose of it, and they immediately no matter where they're given that odorant across a large landscape, they fly south. So it seems as if they have they have learned there is this one-dimensional gradient of this odorant, and home is south of the high concentration, so therefore
But how can they use the odor itself as a directional cue? Because it's transported by wind. If there's no wind or there's no polarizing, sort of alternative cue for navigation, I don't see how the odor itself can actually direct navigation unless there's visual or rheotactic or some other component involved as well.
Well, I mean, it's rheotactic, and I think that's what people are arguing. And Wallraff has argued that there's actually, he's measured the stability of pollutant distributions in the atmosphere, and arguing that the landscape actually, it's not just an odorant around a tree, but there's actually a forest. There are prevailing winds, and so there are actually traditional navigators, know that winds go in different directions at different seasons, and they come with different odors. People in the south of France know that this is this is the wind that comes from Morocco, and we know because it smells like the desert. And I mean, I think there are these large landscape fields. That's what Wallraff and Poppy have been arguing, and of course the problem, and I'm not trying to sidestep that, is turbulence, and are they stable, and how stable are they? And I would argue is that there is evidence, for example, where this has been studied a lot, crayfish moving towards odorants in water of different turbulence. And they actually, marine animals are covered with antennae and sensors, and they're all sensitive to flow, and you can actually increase the turbulence, and the crayfish are better oriented.
Crayfish, I mean, I'm involved in that work.
Oh, okay, yeah.
The crayfish are very unusual compared to most marine animals like a blue crab or others. And that turbulence does extinguish the signal in increased turbulence, and then that in itself reduces the responsiveness or the ability of the organism to navigate. And also, even with respect to turbulence, there's with something like a crayfish, if you look sort of at navigational skill on the y-axis, and maybe it's on the, and then what would be on the x-axis? Some turbulence.
Yeah.
It would be an inverted U shape. So up to some point, it
Yeah,
would perform
yeah.
better, and then it
Yeah,
would
yeah.
drop off.
Yeah. No, I mean,
an optimum?
I mean, obviously, if you've complete chaos, no one's going to orient to anything. What I'm proposing is that animals have derived ways of encoding some kind of moderate turbulence and be able to decode odorants. That's all.
And
Yeah.
so the nature of, let's say, I'm sorry, then I'll finally drop it. I apologize.
No, this is
I
the
just
key
wanted to get
question.
this
No,
straight.
I'm really glad
So
you. Yeah.
the way that I think we view it is that physics imposes a constraint.
Yeah.
And that would deal with advection and turbulent mixing. And the problem is so difficult for macroscopic organisms that they have converged onto a similar mechanism, one that you're describing, that stretches across many different phyla. So there's a physical constraint or environmental
Yeah,
constraint,
yeah.
let's say, that's so difficult to solve that it ends up in simple rules of thumb using an olfactory sort of gating system with a rheotaxis or some other kind of taxis, then ends up being the dominant navigational mechanism.
Well, I would just argue that we don't know enough to know whether or not there is more actual mapping going on, and actually, let me keep going, because actually the next point is what's, again, this is very speculative, but one thing about olfaction is that there's two ways that very disparate taxa perceive odorants the same way. And the first is that perception shifts with intensity, so that one odorant that is, this is documented in humans and in locusts and honeybees and rats, that as you increase the intensity of an odorant, you get this qualitative shift in the percept. It starts like anise and ends up like banana. That's one, and it's interesting because it's found in so many different taxa. And then the other is that how you perceive an odorant mixture depends. It isn't just what hits your nasal epithelium. It depends on your training history. And again, this is, you can either detect and decompose a mixture into the elements, or you can you see it as what Wilson calls an odor object, and that you can't decompose it. And so it's kind of like you see either fruit bowl, or you say you can decompose it into the different elements. And what's remarkable again about these two strange psychophysical phenomenon is that it's found across these different taxa, which of course have very similar olfactory systems. So it seems to be some kind of functional characteristic of that system. And so, but if we are going to examine the idea that olfaction actually functions, it's actually functioning not just chemosensory, but it's chemosensory and integrating with mechanosensory information to make some kind of map of the environment, then these two rules would be very useful. One is that you can imagine that if you've got an intensity gradient and you can map it, then high and low concentrations are going to be demarcated. And if you've got orthogonal gradient, just two odorants, you theoretically could actually break it up into different olfactory spaces. Well, this would be the olfactory space, which is what is being created in the mind of the beholder, and these kind of what I'm calling neighborhoods. So that's just the first rule. That could give you some spatial resolution. And then the second one is that because of the way odorant plumes come together, that if there is an area of stability where two odorant plumes come together, then where they come together, they're going to be they'll be characterized by different ratios. And those ratios could be perceived as a synthetic object. If you were looking for that, it could be perceived as a synthetic object, or it could be perceived as a mixture of elements. And so that just two plumes could be either expected and learned and detected as positional cues, or they could be the XY positions on a Cartesian grid. And that just follows from this, like again, the psychophysics of how animals perceive odorants. So what occurred to me is that this could actually create this parallel map structure that the hippocampus seems to be proposed, the hippocampus is using. And so you could, with just two odorants, you could actually create a cognitive map structure which would allow you to recode odor objects into a common coordinate system and allow shortcutting. And so that just the percepts of olfaction could give you the machinery you need to solve the cognitive map. Okay. So this is where I stand, is let's play with this idea and see how far it can go. And that's really, like I said, I've only been studying thinking about this for about a year. So, but it has some obvious predictions. Well, the first is that, let's think about olfaction differently then. Let's think about it as this actual mapping of odorants in a fluid medium in relation to self-movement, somehow controlling or some controlling for turbulence or working in areas with low turbulence. And if that's true, then the olfactory bulb should actually co-vary in size with demand for cognitive mapping. And that specifically means not just how big your home range is, but how much you need the demand for the extrapolation of novel routes across a home range. Really, the bearing map function. And it's really the bearing map which is the essential ingredient necessary for solving the cognitive map. That's really all you need. So, to foreshadow, really, if you have an olfactory bearing map, you should be able to do a cognitive map. And so if this similar olfactory system allows you to make an olfactory bearing map, then that would be explanation for why arthropods could do the same kind of cognitive map function, even though they don't have a hippocampus. That's where I'm going to go. So you have the bearing map, and then of course, if you can add the other objects, co-encode them, you can get the cognitive integrated map. But right now, the cognitive integrated map appears to be that kind of, the best evidence really is just in birds and mammals. But bearing map type behavior is found also in reptiles and arthropods. Okay. So the prediction is that this olfactory system, therefore, should reflect navigational demand. And there are studies of the relative size of the olfactory bulb in vertebrates, correcting for brain size, correcting for phylogeny. Very careful studies have shown the olfactory bulb increases, and these are just natural history, in some ways, observations. All studies conclude that this is what we found, and we don't understand why discrimination should be so important in this ecological context. But in carnivores, for example, John Gittleman showed that home range size in terrestrial carnivores co-varies with relative olfactory bulb size. There's a lovely study of predatory dinosaurs showing that the more predatory theropod species have larger olfactory bulbs. Perhaps they're ecologically similar to terrestrial carnivores. It's been shown that secondary nocturnal birds, there's been independent evolution of nocturnality in birds, and Sue Healy has shown very nicely that that's associated with larger olfactory bulb, which might be used because they're navigating through olfactory fields rather than visual cues at night. Could be. And finally, homing pigeon. Like I said, the homing pigeon has a larger olfactory bulb than the non-homing pigeon strains. But the best work about it, and in all of these studies, in all cases, it's interpreted as indiscrimination, except for the Poppy and Volleroff camp, which have always seen it as navigation. But what's more intriguing is Barbara Finlay and Richard Darlington's now large opus of work looking at what explains how the mammalian brain evolves, and how can you explain the differential size of different structures in the brain. And what they've shown is that now in mammals, and now they've done it in cartilaginous fish, so kind of the basal vertebrate, that there is the olfactory system really scales alone. And what they found for 97% of the variance of the size of a brain structure of other structures in the mammalian brain can be explained by a very simple model of concerted evolution. And this is, concerted evolution is when all the bits of the brain get big together. There's obviously, that can't be true. Different species have differences. There is a fudge factor here that accommodates any observed differences that we know about. But in general, if you're looking at all brain parts of all mammals, it's the data very neatly are explained by this concerted evolution. But what's been puzzling since '95, when they started demonstrating this, is that the one system that will not follow this rule is the olfactory limbic system. So that's either the olfactory bulb, that was the first demonstration. Then they realized you could just look at limbic system, so olfactory bulb plus hippocampus plus septum, so that whole hippocampus, septum, that whole bearing map circuit, and olfactory cortex. But basically everything that I'm proposing is involved in this kind of olfactospatial navigation system does not scale with the rest of the brain. And they've got beautiful studies to show this. And in fact, the things that don't scale are the olfactory bulb and hippocampus there. And so again, why this variation? None of the papers, they've got every paper has a different explanation. I just spent a week with Barbara talking to her about this, and she agrees that it's a perfectly good hypothesis, is that the reason olfactory bulb is so variable, the olfactory system is so variable across mammals, it appears to be under directional selection, and what we were missing was the selective pressure. And there's no obvious discrimination, just if it's breadth of foraging foods, of diet, things like that, that would explain this. But what I would propose is that perhaps navigation can. And Barbara did a beautiful study, Richard Reep and Barbara and Richard Darlington, looking at the kind of push-pull of the limbic system and the isocortex, neocortex. And what they found is, in general, when you have a neocortex is increasing, you're having the limbic system is decreasing across all mammals. But if you look at it, for example, simians, highly visual, less olfactory. But if they did a second analysis where they looked at what's the size of the limbic in relationship to the brain core, which is everything but limbic and isocortex, and then they looked at that, they come out with these different patterns. And again, I sound snarky here, but I'm kind of I guess, evangelist now for olfaction. And I looked at this paper, and they didn't have any explanation for this beautiful, beautiful data, which was this, where this is the relative isocortex size on the x-axis, relative limbic size on the y-axis, adjusted for core. And what you can see is these are different taxa. So this is simians. These are prosimians. This is echolocating microcropter in bats. These are insectivores, shrews, moles. These are terrestrial carnivores or carnivores. And so it's clear that there is clustering by taxa for this, in addition to this general pattern of one gets bigger, one gets smaller. And if you think about it in terms of their predatory strategy and how they're using olfaction, I would suggest that maybe we could break these animals up, break the behaviors down into two foraging strategies, a kind of detector strategy where an animal is just going out there and using its sensory ability to, it's mapping space crudely, but it's using its, then it goes out and detects prey, either because the prey are either random or they're so obvious you don't need to predict where the prey are, you're just detecting them. You want to, the star-nosed mole is the ultimate detector. A predictor strategy is that prey actually can be, there are patterns in their spatio-temporal distributions that can be outwitted. And if you can keep track of these patterns, then, which I would argue does require greater neocortex, isocortex commitment, then you can actually, prey on them more effectively. So if you're predicting using odorants, then you should not only have a large isocortex, but you should actually maintain this large limbic system for this olfactory mapping. But if you're using another sensory ability to map, then you could decrease the limbic size. And if you look at the data, here's some examples. So you've got olfactory, non-olfactory, major sensory inputs, detectors and predictors, and that kind of that does explain some of these patterns, or it's a hypothesis that might explain these. Is that you've got something like an animal eating small scattered insects, like a shrew, which has a high limbic, low isocortex as a spatial, an olfactory detector. Whereas a non-olfactory detector would be a microchiropteran bat, an echolocating insect-eating bat, where it has replaced spatial olfaction with spatial audition, but it's also mostly a detector, not trying to predict the location of flying insects. These are gross generalizations, obviously, I realize that. And a predictor would be something like a terrestrial carnivore, like a wolf, which is both mapping its environment in terms of odor, so it needs this large limbic olfactory mapping system, but it's also predicting the movements of its smart prey, so it's got its large isocortex. And a simian would be spatial audition has supplanted spatial olfaction, and so you've got a high isocortex and a low limbic. So that's just a suggestion that we could take data from REAP and et al. And put it through the olfactory navigation filter, and then see and come up with new predictions and possibly new interpretations. Another interesting thing is that adult neurogenesis is a very interesting phenomenon because the new neurons being born in vertebrate brain are actually found throughout vertebrates, in many places throughout the brain. But in mammals, it's very interesting. It's limited to two places, the olfactory bulb and the hippocampus. So what we can say is and that's true of all vertebrates. So there's only two locations in all vertebrate brains that have adult neurogenesis, and that is the granule layer in the olfactory bulb and the granule layer in the hippocampus. And these, So one hypothesis is that neurogenesis, which we know increases with spatial enrichment and movement, that it is actually a signature of olfactory spatial mapping. And we're just beginning to get more and more data on rates of neurogenesis in different species. Microchiropteran bats, echolocating bats, was the first species to be discovered that they have small olfactory bulb size, and they have the first mammal to show no or low or absent hippocampal neurogenesis. It was thought that this was mouse, rat, human, and it was just general, therefore. And it turns out that Microcoroptera in bats, it's very variable, and it's absolutely, and it's clearly absent in some species. In contrast, Megacoroptera in bats, fruit bats, have intermediate olfactory bulb size, and now a new study just out a year ago has shown, in fact, they have intermediate levels of hippocampal neurogenesis. So, and olfactory and fruit bats have limited echolocation abilities. They're not as specialized. They have very long, large eyes, large nose. They're very different, completely different sensory world than the specialized Microcoroptera in bats. But this suggests that adult neurogenesis is actually not just about hippocampal plasticity, but it's specifically orchestrated with the olfactory information into making some kind of olfactory map. So finally getting back to the invertebrates, again, the convergence morphology, no one's denying that. More recently, there's a very interesting study of a marine annelid worm by Detlef Arendt's group showing that there is a homologous gene network that underlies the development of the mushroom body in this marine annelid, which they argue is a better model for looking at our common ancestor vertebrates, protostomes and deuterostomes, vertebrates and invertebrates. And what they found by using new methods to line up to put in register the sequence and the expression of genes expressed during the forebrain development, they found homologous networks for the mouse forebrain, the mouse pallium, and the mushroom body or the forebrain of the associative big brain of the worm. And they are suggesting that this is evidence for a single brain. The brain evolved once and in bilateria, and it's based on these genetic evidence. There's possible evidence from arthropods from Nick Strausfield's work, where he's been looking at the relative size of olfactory structures and the mushroom body. So you can think of this as the antennal lobe is the equivalent of the olfactory bulb. The mushroom body is the equivalent of the hippocampus. And what he's shown is that you don't, although the mushroom body was thought to be purely olfactory, there's a species like the water strider, which moves around the water and has reduced olfactory structures. Instead, it's getting mechanosensory information from what it signals and communicates by water ripples between other water striders. And that, in fact, it's kind of similar to the Microcoroptera in bats. It's lost olfaction. Instead, it's using this mechanosensory modality, and it's been a real puzzle. Why would they have this large mushroom body, yet they have they have very small antennal lobes, because the mushroom body is supposed to just do olfaction. And so, again, wondering if this, like the bats, this is a secondary switch from odorant olfactory field mapping to another mechanosensory field mapping. And there's many cases that Nick's just published an 800-page book on the arthropod brain, and continually is bringing up this question: what does the mushroom body do? The size of the mushroom body cannot be explained by just olfactory discrimination, but I think it's very consistent with the idea it's actually mapping. So if this is, so what I'm really jumping off a limb here and saying that I kind of agree with Arendt and Strausfield that there could be one brain that evolved from this olfactory architecture. This is why we're calling this cog evo devo. You've got to come up with a bandwagon, right? But that's actually Darwin's notebook, his first tree that he drew, and proposing that the bilaterian brain could have evolved from this one gene network we know, in aqueous chemosensory world. that this simple parallel map structure could be used to become a much more effective predator, then this, many people have suggested Cambrian explosion could have been cognitive, that Avia Blanca, that it was the appearance of associative learning that led to increased success and therefore increased body size and increased predation and increased, adaptive radiation of all the animal phyla. I would just tweak that and say, what if it was in fact olfactory spatial associative learning that was evolving using a parallel map structure, and that's why we find this structure in the hippocampus and the mushroom body, because it could be obviously convergent, it could or it is it's a homologous common ancestor. So in conclusion, I love this quote from this beautiful painting by Ken Rode, and of the Cambrian world. The act of smelling anything is remarkably like the act of thinking itself. I could not agree more. I think that we should, other people have pointed out that olfaction is so difficult that because it was the first problem the nervous system had to solve, it channeled our nervous system and cognition, learning, and brain in a certain direction, because we solved the hard problem first. Vision was easy after that, but because of that, we became the brain. We have the brains that we do. There's no question that olfaction is a tough nut to crack, and so. In summary, this is the hypothesis that it was chemosensory navigation was ancient, universal, that this bootstrapped the ability to extrapolate across space. It started, again, with very small animals which were close to the surface, which were not possibly not having to deal with much turbulence until they became more successful, larger, and at that point you're getting other sensory modalities, particularly vision, emerges very early, Precambrian, and that would have bootstrapped this small olfactory bearing map would have bootstrapped then a much more robust and successful multisensory using vision and magnetic fields and all the other things we know are used for navigation. And then that would address problems in turbulence, and that this is why there's directive selection has continued to this day, which is why olfactory systems scale differently, evolved independently, and it's still critical for navigation in most animals, even birds, possibly driving these patterns we're seeing of adult neurogenesis. And because all of this is all being built on this foundation of this this kind of archetypal cognitive map based on olfaction. So I'd like to thank the many people, Randolph Menzel, Leslie Kay, Rachel Hers, Corey Bargman, who are all experts in olfaction who have been teaching me, and Neil Vickers, Wayne Potts also, and my other collaborators, Mimi Cole, Bob Full, and Dan Kotichek and CJ Taylor, who are engineers interested in movement, and my students, and funding from NSF. Thank you very much.